Hi dear watchers! Today, we are going to run through the laboratory experiment that we did for our first semester in Laboratory Entomology at Far Eastern University. We will discuss about baiting and catching insects, pinning and mounting the ants that we have collected, identifying the species collected through dichotomous key, and mosquito identification. The first experiment conducted by the group is all about baiting ants and catching arthropods using different equipment such as bait with jar and proteins and sweeping net and the kill jar. During this experiment, we laid traps in different areas of our university and waited for 30 minutes to gather the trapped species. We also caught moths, mosquitoes, and flies using a sweep net.
The materials used for this activity include entomological pins, paper tips, a Gucci ball, tight bond, which is the water-based glue that will be used for fixing the specimen on the paper tip, entomological probe, which can be improvised with barbecue sticks and entomological pins, and lastly, a pinning block, which we were able to acquire from our professor, Dr. Pagong. For the pinning of the specimen, which in this activity is ant, a paper tip was first properly pinned on an entomological pin with the aid of a pinning block. This part is crucial as the paper tip will be used as the stage of the specimen for proper viewing and observation of the specimen's external morphology.
The mounting process begins by gently drying the specimen with a tissue to remove any excess moisture. Next, under a dissecting microscope, carefully apply a small amount of glue to the tip of the paper stage using a probe. Use just enough glue to stabilize the ant, avoiding excess that might affect viewing its morphology. Moisten the probe with water to help the ant adhere to it, then transfer the ant to the paper stage. Position the ant with its dorsal side facing up and the head pointing to the left. Ensure the paper tip is placed between the first and second coxa, securing the ant in a stable position for mounting.
Dichotomous keys are used to identify the mounted ant species. These keys present a series of paired statements or questions allowing researchers to progressively narrow down the possibilities based on the observed morphological features. We started off with a collection of ants using both active and passive collections and then mounted the ant species. We then carefully examined the specimen and selected the appropriate options so one could accurately determine the species. Furthermore, accurate identification requires precise observation and the use of a clear and specific dichotomous key.
As we examined the ant, we assessed whether the specimen possessed or lacked the morphological characteristics described in the key. Using this method, we identified the ant as belonging to the subfamily Formicinae as it lacked a petiole and a sting. Additionally, it had an acidopore at the apex of the gaster, and dentinal sockets were situated behind the posterior margin of the clypeus. Based on this information, we proceeded to identify its genus and concluded that it belongs to Ochophylla. This is because the specimen's antennae were divided into 12 segments, its mandibles were triangular with more than 10 teeth, and they were shorter than the head length. Additionally, its eyes were large but did not take up most of the head. Further research also led us to identify the species as Ocophila smaragdina, also known as the Asian weaver ant.
Our group has managed to capture at least 15 to 13 mosquitoes around streets of Spain. The mosquitoes were transferred into the killing jar for proper preservation and collection. The collected specimens were eventually relocated into a glass presentation for proper observation. Hence, with the use of the septum microscope, we were able to obtain different identification of the species through its morphological form. In this activity, we were able to determine the kind of genus through in-depth analysis while viewing the specimen. The probe was a tool that aids in separating the significant parts for helping the determination of the specimen.
To determine the sex of the mosquitoes, we examine the antennae. Males typically have bushier, feathery antennae, while females have thinner, less conspicuous ones. Identifying the genus involves assessing key traits such as the length of the palps and the shape of the proboscis. For instance, Anopheles mosquitoes have long palps equal to the length of the proboscis, while Aedes have shorter palps. This is according to surveys in 2012. In Culex mosquitoes, the palps are also short in both sexes, but males have relatively longer palps compared to the females. They can be recognized by their blunt tip abdomens and lack of distinct markings, differing from the Aedes, which often have white prominent patterns. Additionally, Culex mosquito exhibited scaled wings without the spotted patterns found in Anopheles. This is according to Becker et al. In 2010. Accurate genus and sex identification is essential for understanding the roles that these mosquitoes play in disease transmission, particularly since female Culex are vectors for diseases like West Nile virus and Japanese encephalitis. This is according to Centers for Disease Control in 2020. In summary, the 4 laboratory exercises provided an in-depth exploration of medical entomology, emphasizing both technical skills and scientific knowledge. The baiting and catching experiment taught us methods of insect capture and highlighted the ecological importance of monitoring insect populations. The pinning and mounting exercise demonstrated the precision required for specimen preparation, ensuring the preservation of important morphological features for viewing. By using the dichotomous key, we gained proficiency in the systematic identification of ants, honing our observational skills to accurately classify species. Finally, the Mosquito Identification Lab offered insight into the distinguishing features of mosquito species, emphasizing their role as vectors in disease transmission. Through these assessments, we develop a solid understanding of the processes involved in identifying and studying medically significant insects, which is essential in managing public health risks and associated with vector-borne diseases.